las personas, los que están aquí presentes, por favor, si quieren los subtítulos en español, en la barra de herramientas, por favor activar los subtítulos y elijan español como el lenguaje preferido. Okay? Okay. So you have the floor, Mr. Dr. Thomas Cech.
Thank you very much for the nice introduction. I am happy to join you today from Colorado. It would be much nicer to be in Peru personally, but we will make do with this today. And I am going to start by sharing my screen because I have some slides to help me talk about this topic of the ribonucleic acid, or the RNA. We had some recent exciting results in my laboratory, and so I have changed the topic of my talk, and after a bit of an introduction, I will move to some recent work which I hope that you will find of interest. And so the topic is the magic of RNA. What can RNA do beyond being a message? So when I talk to non-scientists about RNA, I usually start with its mother molecule, which is the DNA double helix, because most people are generally comfortable with DNA. They know that it is responsible for our inherited traits, or mutations in the DNA can cause genetic diseases. We can trace our family history through our DNA, and it can be even used to solve crimes. But when we move from DNA to its daughter molecule, RNA, even though you can tell by the letters, they both are nucleic acids, the RNA is a single-stranded copy of one of the DNA strands, and even though it contains the same information as one of the DNA strands, people think that it is very complicated. And so I have been trying to explain to people that RNA is really very easy to understand. In fact, we have known for 60 years that the code carried in the DNA is used to copy into an informationally equivalent molecule of messenger RNA, and then groups of 3 of the informational units along the RNA, we call these bases or nucleotides, are used to specify a particular amino acid laid down in a protein chain. And the proteins then do fantastic things in all of living systems. For example, in humans, they are responsible for our muscles moving. They are responsible for the enzyme in our stomach digesting our food. They are responsible for our heart beating and our neurons functioning. So each of these fantastic proteins has a gene made out of DNA that encodes it, and the RNA is the intermediate in this process. Now that the mRNA has been used to make a vaccine that saved an estimated 10 to 20 million lives in the very first year that it was introduced in 2020, people are now more familiar with this messenger function of RNA because it can be used to train the human immune system to be on the lookout for a particular protein, in this case, the spike protein that coats the outside of the SARS-CoV-2 virus. So if this were all that RNA could do to be this fantastic messenger, this might be enough to call the 21st century the age of RNA. But in fact, RNA can do much more than this. Beyond being a message, non-coding RNAs, which do not do anything in terms of specifying a particular protein, serve important functions in all living forms of life. I will talk about only 3 of these today, although I would love to talk about 20 of them if I had enough time. One category is that RNA can be a catalyst. We call these ribozymes, ribo for ribonucleic acid, and zyme for enzyme. But more recently, we have found that RNA can regulate the expression of genes, and it does this by regulating 2 protein complexes, one of them called DNA methyltransferase, or DNMT1, and the other one called polycomb repressive complex 2, or PRC2. And the detailed mechanism of this inhibition at the atomic level is something that the people in my laboratory have been able to figure out very recently. So let's start then with RNA being a catalyst. This is an older story, the ribozyme story. And when I moved to Colorado to start my own laboratory, I started working on pond scum. Now, why would anyone work on pond scum? Well, it wasn't just any old organisms in the pond. It was this ciliated protozoan, a single-celled animal called Tetrahymena thermophila. And I started working on it because it contains 10,000 copies per cell of a gene that makes RNAs that form part of the protein synthetic machinery. And this is a lot of material to work with because in most of our cells, genes are present in only 2 copies, one inherited from mom, one inherited from dad. So if you are a biochemist and want a lot of material to work with, 10,000 is a real advantage. What we found very early on studying this gene was that it made an RNA molecule that underwent an RNA splicing reaction. This is a cutting and joining reaction to remove an internal portion of the RNA, which was interrupting the sensible, useful portions of the RNA molecule. And this was happening in the test tube very efficiently and at the same sites that it took place in the living cell, which made it of great interest to us. But the RNA seemed to be doing this reaction without adding any protein enzyme to catalyze it. Could it really be that the RNA was splicing itself? This seemed extremely unlikely because ever since James B. Sumner gave his Nobel Prize lecture in 1946, everyone knew that all enzymes are proteins. So we were quite confused for some time, trying to find some hidden protein enzyme, perhaps contaminating our RNA preparations. And this leads me to a Christmas party in our chemistry department, at which time my graduate student, Paula Grabowski, presented me with this Christmas gift. Was there a protein hidden in our reactions? Or was there not a protein, in which case the RNA must be having the catalytic power within itself? And that turned out to be the case, that the RNA by itself was the catalyst. We were able to show that in 1982. Now, this was of great interest to another group of researchers who were not studying Campbell's primordial soup, but instead the mixture of elements that was present on the primitive Earth some 4 billion years ago, when the first self-reproducing prebiotic life form first is thought to have arisen through random chemical processes. And when scientists thought of how this could have happened, they were quite skeptical, because to have a self-reproducing system, you need to have an informational molecule. In our cells, this is DNA, the double helix, but DNA doesn't do anything by itself. You need to have a protein enzyme to copy that DNA to pass it down to the next generation of the organism. So how could everything get started if you needed, in the same tiny little droplet of water at the same time, through random chemical processes, an informational molecule and a functional enzyme molecule? Well, now that we had found that RNA had both information capacity and function, it could be its own copying machine, as well as being the molecule that was being copied. And this is the so-called RNA world hypothesis for the origins of life on Earth. An exciting possibility, one that is scientifically plausible, but since it relates to a historical event, it is difficult to be sure exactly how the first self-reproducing molecule began on the primitive Earth 4 billion years ago. Nonetheless, this interest in the catalytic capabilities of RNA led rather quickly to the Nobel Prize in Chemistry. My daughter, who was 7 years old, was allowed to attend the ceremonies, and my 3-year-old daughter, who is now a PhD in cell and molecular biology, was too young and was not allowed to come along for the festivities. How can RNA have this sort of activity? Well, it folds up. I said it before that it was single-stranded, but that strand can fold within itself into a limitless number of complex shapes. And some of these catalytic RNAs, shown here 3D printed by my colleague, Professor Deva Satyaputra, in Vancouver, British Columbia, you can see the wondrous capabilities of these folded RNA molecules. And before I move on to the recent results from my laboratory, let me just say that there are a range of RNAs that are intimately involved in catalysis that have been found after the original discovery. In some of these, starting on the left, RNA is both the active molecule and it is sufficient for catalysis. In the middle are some cases where the RNA is, of course, the active component, but it needs a protein or protein helpers in order to do its job. And then on the right, we will have a variety of RNAs where the RNA is not catalytic, but it is still essential for a biological process. So let's look at a few of these in a little more detail. On the left, you can see 3 examples of folded RNA molecules, now shown just in a 2D, two-dimensional representation, rather than a 3-dimensional representation that I showed on the 3D printer. On the left, we see the tetrahymena, catalytic RNA, the ribozyme, a much smaller catalytic RNA called the hammerhead, a quite tiny 45 nucleotide RNA published just this year by Philip Holliger at the MRC in Cambridge, England, that can copy itself and copy a template of itself, a big step towards RNA self-replication in the test tube. But then there are cases such as in the middle, and the bacterial RNase P, ribonuclease P, was discovered by Sidney Altman, who shared the Nobel Prize in 1989. And it has a very small protein that collaborates with the RNA to help it fold. But the RNA is the catalytic entity of this complex. Then further to the right, we see the ribosome, which binds messenger RNA and transfer RNA and catalyzes protein synthesis. This is, again, an active RNA component. The RNA is shown in the cyan and silver colors, but the proteins, dozens of small proteins, serve as helpers to help the RNA fold into the correct shape for catalysis. Now let's move on to cases where we don't need to look at those, and we'll move on to cases where the RNA is not catalytic, but it is essential for the process. And this is telomerase, which extends the ends of our chromosomes. The catalytic subunit is a protein, telomerase reverse transcriptase. The RNA serves as a scaffold to bring that protein and several others into the complex, and it also serves as a template for DNA synthesis at our chromosome ends. The top right example is a small interfering RNA. These have been converted into a variety. They are found naturally, first found in worms, but found now naturally in humans as well. But synthetic copies of these RNAs can be used to cure diseases by disrupting disease-causing RNAs in our bodies. And this is, again, a case where the RNA serves as a guide, but the cutting activity that inactivates the unwanted messenger RNA is found in a protein component, part of this RISC complex. And finally, at the lower right, we see the famous CRISPR genome editing, which was the source of the Nobel Prize to Jennifer Doudna and Emmanuel Charpentier in 2000. And this, again, has an essential RNA which helps the complex find where in the chromosome it should do its gene editing, but the actual cutting of the DNA is done by a protein partner called the Cas9 protein. So these are just a few of the enormous number of examples where RNA plays critical roles in catalysis. And going from left to right, we see the increasing importance of proteins in the reaction, not important at all for the pure ribozymes shown on the left, and increasingly important as we move to the right. And it is interesting to think that perhaps this is also the evolutionary trajectory. Maybe the ancient RNAs didn't need proteins, but once RNA learned how to make those proteins, which were useful to it, then the proteins took on partnership opportunities with the RNA, and we move from the left towards these systems shown on the right. Now, that was a very quick tour through the tremendous capabilities of RNA, but I would now like to turn to some recent work from my own laboratory here at the University of Colorado, Boulder. As I have said, and as you all know, DNA is used to copy into an RNA copy, which is used to direct the synthesis of particular proteins. But wouldn't it be interesting if the RNA could also reach back and either positively or negatively affect the production of itself? If it could regulate the ability of the DNA to be able to be copied into RNA. And why would we come up with such a crazy idea? Well, it's because many DNA binding proteins that in the textbooks are said to be DNA binding, and if the students were given an examination in a class, the correct answer would be that these proteins bind DNA. They also bind RNA. And they often bind RNA with a higher affinity than that which they use to bind DNA. And these include transcription factors, and what I'm going to talk about today is DNA methyltransferase number 1, DNMT1, and PRC, polycomb repressive complex number 2. The people who did this work are my colleagues, postdoctoral fellow Jiru Song, shown on the left, 2 graduate students, shown in the middle on a laboratory hike in the beautiful, always sunny mountains of Colorado. This is Jess Song and Ella Tomer. And the work I'm going to show today would not have been possible without a wonderful collaboration with a new faculty member, Vignesh Kasanath. And so the first of these topics is then that RNA can regulate DNA methyltransferase, DNMT number 1. So why would DNA be methylated? You all know what a methyl group is. It's just a carbon and 3 hydrogen atoms, a very small addition to the cytosine nucleotide along the DNA. And this occurs upstream or preceding many genes whose expression can be regulated at the level that we call epigenetics. Now, what does epigenetics mean? Well, it means at some level above the genetic level. So even without mutating the DNA, without changing the code of the DNA, a gene can be turned off, as shown on the bottom diagram by that red X, by methylation of nucleotides upstream from or preceding the start of the gene. And this is epigenetic gene silencing, and the enzyme that adds these methyl groups is the DNA methyltransferase, a huge protein whose catalytic domain is the pink domain on the right-hand end, and all of the boxes before that represent regulatory domains of the protein. And the reaction catalyzed by the DNA methyltransferase is to, after DNA is replicated, one of the 2 strands, the daughter strand, will not have its methyl group anymore, and the DNA methyltransferase, by performing the reaction shown here, restores complete methylation at the 5 position of the cytosine base. Now, the biology of this system was figured out about a decade ago by John Rinn and Dan Tenon at Harvard University, that RNA can inhibit or turn off the activity of this DNA methyltransferase in living cells. But how did this happen? What was the mechanism? What were the structures involved? And so here's just some biochemical data showing that in the presence of a particular sequences of RNA, such as repeats of G and U, the DNA methyltransferase is completely repressed. It's turned off. Whereas other RNA sequences of the same length, such as 40 A nucleotides in a row, have no effect on the DNA methyltransferase activity. But what is the mechanism then? So by using cryogenic electron microscopy, a rather new technique that allows one to see individual atoms within a protein RNA complex, Jess Song was able to determine the structure of this RNA bound, this GU-rich RNA bound to the protein. And it did not turn out to be colored blue in the experiment because this is just electron diffraction, but she colored it blue here so that it's easy for us to see. And it is superimposed on the model of the protein where you can see internally within the protein all of the amino acid strands that make up the protein. So why would this particular binding of the RNA to the protein be, let's move ahead here. Okay, and so she took that extra density that was on the surface and just pulled it away from the rest of the protein. And it has this rather interesting four-armed sort of shape. And when she took the folded form of the UG repeat RNA, a structure that was determined by Sam Butcher at the University of Wisconsin, and then rendered that structure at the density of 8 angstroms so that she could see what it would look like in her cryogenic electron microscopy structure, she determined that it should look like this yellow sort of pillow-like structure. And sure enough, that is the same shape and the same size, pretty much. The RNA that we used for the experiment was a little different in length than the RNA used by the Butcher laboratory for the NMR structure, but nonetheless, you can see that they overlap rather nicely. And so how does this then explain the ability of the RNA to prevent the enzyme from working? And here we superimpose the structure of the RNA bound to the protein with the DNA bound to the protein, which does not occur in the same complex. You can see that the blue RNA is bumping against the yellow DNA double helix. They cannot both be there at the same time. So this in a very simple way, explains how the RNA prevents the activity of the DNA methyltransferase. If it cannot bind to the DNA substrate, it cannot perform a reaction on it. Well, when we move from that system to the different epigenetic silencing system, we see that RNA regulates this other complex of proteins, the polycomb group of proteins, in a very different way. It's not a simple blocking of the place where DNA needs to bind. It is intricately engineered for the RNA to be inhibitory. We did not predict this. We were just set out to find the structure of the RNA bound to the protein, much as with the DNMT1 that I just showed you. And the answer that we obtained was really quite amazing. So first, before I show the structure, let me just explain what it is that the polycomb repressive complex 2 and its colleague, polycomb repressive complex number 1, both are enzymes that change the nature of the histone proteins that wrap up the DNA into the chromosomal fiber. And when they put these methyl groups or other chemical modifications on the ends of the histone proteins, it causes the histone proteins to condense the chromosome fiber into something we call heterochromatin. And then RNA polymerase, which has to copy the gene into RNA, is unable to get access, and so the gene is turned off. This is absolutely critical for human development, for example, in the regulation of the so-called Hox genes. It is part of the reason that one of the 2 X chromosomes in females is inactivated, not mutated, but simply converted into a tightly compacted ball of chromosome to give the important consequence that females have 1 active X chromosome And males have one active X chromosome. And since there are so many important genes on the X chromosome, it's critical that females and males have the same number of active chromosomes. And again, polycomb repressive complex 2 helps inactivate one of the two female X chromosomes. And this is also so important in cancer that there are now approved drugs that inhibit polycomb repressive complex 2 and prevent it from aiding tumor cells to do their job. And so what is it in DNA that in RNA, talking about RNA here, what is it in RNA that PRC2 actually recognizes and binds? Well, it's very simple. It's either runs of the G letter of the alphabet of RNA. Remember, RNA has 4 letters in its alphabet, A, G, C, and U. If those G's are unstructured, then the polycomb repressive complex 2 can bind them, and then somehow it gets inhibited by binding them. If there are 4 of those blocks of G's, they fold up into this beautiful structure called a G quadruplex, and that is a particularly high affinity binder and good inhibitor of PRC2. Whereas if these G's are Watson-Crick base paired into a stem loop, they are not recognized by polycomb repressive complex 2. And so this inhibition of PRC2 by RNA was discovered by Jeannie Lee's laboratory at Harvard University and Danny Reinberg's lab at NYU in New York City. A couple of years later, Richard Jenner's lab in London and my lab figured out that the RNA was blocking the activity of this enzyme by preventing it from binding to the DNA. So if you can't bind to the DNA, you can't find the histone proteins that need to be modified by this silencing enzyme. So the biology here is that if a gene is active and making RNA, that RNA will have some groups of G's in its sequence here and there. When PRC2 comes along, it will bind to that, set those G's in the RNA, more or less depending on how many G's there are in the RNA. And this prevents it from acting on an active gene, and it will then, after a minute, be evicted from this complex, and eventually this will drive the silencing complex to genes that have already been transcriptionally silenced, which is a good way to then drive the complex to places where it is supposed to be working. So in order to solve the structure of the PRC2 bound to RNA, Jie Rousong in the lab had to do something very high tech. He made a 2-dimensional crystal of streptavidin, only 1 molecule thick, on an electron microscope grid. And then the RNA that binds to the protein had a biotin on its end. Biotin is a small molecule that has a very high affinity for streptavidin and able to show that the molecules on the electron microscope grid were only there in large numbers if you had both the RNA and the PRC2 complex, as shown on the left. If you have no RNA, very few PRC2 molecules bind to the grid. So this was a way of selecting for RNA molecules which were bound to the, for selecting for protein complexes which were bound to RNA. And he was in for a real surprise when he solved this structure. And this was now solved at rather high resolution. And you can see the in various colors the different protein subunits that make up this complex, but it was twice as big as we thought it would be. There were two molecules that the RNA shown in gold on the upper right brought together two of these PRC2 complexes, and they are touching each other through this protein-protein interface shown in the middle of this molecular simulation. Now, why is it important that the RNA is bringing these protein amino acids together? Because this protein-protein interface contains exactly those amino acids that the protein needs to be a catalyst. When they are involved in this dimer form, they are not free to serve their catalytic function, and thereby the RNA binding inactivates two of these PRC2 complexes by one RNA binding event. Okay, so let me then just go on and say that I'll just go to the lower right here and say that this ability of this arginine is a positively charged amino acid that binds to the negatively charged backbone of the RNA. The ability of this arginine-rich loop to bind the RNA in PRC2 might be a more general hint about how a whole class of these proteins works, because arginine-rich loops have been found by Rick Young's lab at MIT in Cambridge, Massachusetts, to be present in many transcription factors, and these are then also binding RNA, just like the PRC2 does. Well, I'm going to finish up now and just say that what I've said today is that RNA can be a catalyst. It can, for example, splice itself, as shown by the tetrahymenorybozyme. It can regulate DNA methylation in the human cell nucleus, and it can regulate histone methylation. And both numbers 2 and 3 are a way of regulating the epigenetic silencing of genes. And these stories maybe would be enough to allow me to claim the 21st century really is the age of RNA. And I have written a book that talks about, for non-scientists and also for mathematicians, other smart people who have not had training in biology, about how this fantastic molecule RNA works. And it has been translated into Spanish. It is also available in English. Am I making any progress in teaching the general public about RNA? Well, I don't know. But here in the New York Times crossword puzzle recently, 28 across was molecule that can self-splice, and the answer, of course, is RNA. I thank you for your attention.
Okay, thank you very much, Dr. Cech, for your interesting and valuable presentation. Now, we will now move on to the panel discussion. I will introduce each panelist, and they will present their questions to Professor Cech. Okay. Our first panelist is PhD degree Martha Esther Valdivia Cuya, Dean of the Faculty of Biological Sciences at the National University of San Marcos, and she will present her questions directly in question. Dr. Valdivia, you have the floor. Are you there?
I'm here.
Okay, go ahead.
Hi, my name is Marta. It's an honor to share this meeting with so distinguished Dr. Thomas Cech.
Good to meet you.
Nice to meet you too. Nice to meet you too. 37 years later, why did you study the RNA of Tetrahymena thermophila. Was an accident choice the species that you choose?
Yeah, so we chose that organism because it had 10,000 copies of the same gene, all making RNA at a very high rate. And so
Yeah.
this gave us more material to work with. It made our studies go faster to work with this organism. The fact that the RNA was self-splicing was, of course, just lucky for us. We did not predict that ahead of time.
Was an accident result.
Yes, I mean,
The same that everything.
it is called serendipity, right? That there's accident involved and good luck, but also the researcher has to understand that it is worth trying to understand how it is working. So it is not just luck, it's a combination of luck and also some skill.
Okay. Due to the RNA is a versatile molecule, may have been an ancient-like DNA from the early stage of air. What reflections can you share with us about that?
Yes, well, as I hope I said during my talk, it is an exciting hypothesis. Many of the steps of this RNA replicating itself have been reproduced under laboratory conditions. But to be honest about it, this does not prove that that is the way that it happened on the primitive Earth 4 billion years ago. So we need to remain open-minded about the answer to the historical question about how did life start.
Thank you so much.
Okay.
Should we go with Luis now?
Yes, we have our second panelist is Dr. Nathalie Flores Fernandez. She's a professor at the National University of San Marcos. She will likewise present his question in English. Dr. Flores, the floor is yours.
Yes, dear Thomas, nice to meet you. It was a pleasure to hear this talk. I'm a professor at San Marcos, and also I work as a researcher at the University of Oxford in the UK. My background is biochemical engineering. So my question is, we know how important were the RNA vaccines in COVID time, as you mentioned at the beginning of your presentation. My question would be, what would be the potentialities or other potentialities of RNA that we are underestimating in terms of biopharmaceutical development, and also how we can manage the acceptance of population, because we know that people is avoiding to use this kind of technologies because they concerned about the side effects, but we as a biologist or biochemists know that the beneficial is higher than the risks. So how, as a scientist, we can manage these trends to avoid this kind of technologies?
Well, you said that last part so well that I think you answered your own question, which is that we as scientists know that every medicine has some risk involved. There's no such thing as a completely 100% safe vaccine or medicine. So you have, but the safety studies that have been done on the mRNA vaccines have shown that it is remarkably safe. You have to give it to millions of people before you find even a small amount of adverse events. whereas getting the COVID-19 disease has a much, much higher risk of adverse events. And so it's a balance between a very small risk from the vaccine and a much larger risk from not being vaccinated. So the other thing I think we have to keep telling the public or share or listen, we have to first of all listen to them to find out what their concerns are. But then I think if we explain to them that we have understood how messenger RNA works for 60 years, this is not a new discovery. This is not something that is untested. It's something that scientists have been very comfortable with for more than half a century. I think that might give some people some confidence in these vaccines. In terms of what could be coming up next, I think a flu vaccine that is much more accurate and therefore much more effective than the current vaccine is already been produced and is perhaps close to being approved for human use starting in our country. If it doesn't happen in our country, I hope other countries will evaluate it for their own public health. And then cancer vaccines, a fantastic idea. We might be able to immunize people so that their immune system fights against their own tumor, is an idea that, again, seems to be just around the corner. I cannot promise that it will be successful, but it is looking good so far. Thank you for your
Thank you for your reply, and congratulations for the Nobel Prize. It's a very, very important milestone, and we are very proud of your knowledge. Thank you so much.
Thank you, Doctor. Okay, we have another panelist, Dr. Cech. Our third panelist is PhD degree Jose Carlos del Carmen, Dean of the Faculty of Medicine at the National University of San Marcos. At this moment, we will share a video with his question, which has been previously recorded in English. Please allow us a moment while we play the video.
Thank you, Dr. Cech, for your excellent conference. Considering that Peru is a country with complex genetic heterogeneity resulting from centuries of history and intermingling, how would you envision integrating RNA-based therapies into personalized medicine models for population with high genetic heterogeneity, such as those in our region?
Well, thank you for that question. And of course, the genetic heterogeneity of a population can affect many aspects of health and disease, including susceptibility to viruses, but also the extent to which inherited diseases are cause either a very severe form of a disease or a somewhat less form of a disease. So I think that when we do the clinical trials on these RNA medicines, we have to be careful not to choose just people who look like me as the subjects for the safety and efficacy trials, but we have to use a broad range of the people from the population, because some of these therapies might be, for example, more effective on an indigenous population than on population that came from Europe, or the other way around, right? It could be that it's less effective. And we just should have this information so that we can give physicians good advice about which patients are likely to benefit from a particular RNA therapeutic.
Okay, we have our fourth panelist. Is PhD degree in Neftali Ali Borja, Dean of the Faculty of Chemistry and Chemical Engineering at the National University of San Marcos. Unfortunately, he's unable to join us live today, so we will read his question on his behalf. Okay, Dr. Cech, and this question is as follows. Dr. Cech, how do you see the role of RNA-based therapies evolving in comparison to traditional DNA-based approaches in terms of precision and safety?
So I think that both will have a role in the medicine in the future. For the moment, the advantage of the RNA therapy is that it is not transgenerational. It is not inherited. It is not copied over. It has not become part of your chromosome. So until we figure out the safety of some of the genome editing technologies, I think the RNA therapeutics will be attractive because they do not change our genes. Ultimately, once we figure out the safety of the gene therapy approaches, they may be more attractive because they do give a permanent change to the chromosome. And if you want to fix a genetic disease, that's sort of a hardwired change in the chromosomal DNA is very attractive. So for the moment, though, I think that they each have a role to play, and they can sort of support each other as therapeutics.
Okay, thank you very much, Dr. Cech, for your reply. Okay, thank you to all our panelists for their insightful questions, and Professor Thomas Cech for his fascinating presentation. And now, it's my great pleasure to present what's the name of this one? Say that, sorry, minutes. Okay.
Hi, Carl.
Okay, to introduce Dr. Maria Natividad Segarra, the faculty, to deliver the official welcome and recognition on behalf of the institution. Dr. Segarra-Garay, please proceed.
Good afternoon, Professor Cech. Thank you very much for your inspiring and insightful lecture. It has been a true honor to learn from your work and experience. We also extend our sincere thanks to our panelists for their valuable contributions and thoughtful discussion. Thank you for being part of this session. My apologies, I experienced a sound technical issue. Good. Distinguished audience, academic authorities, fellow professors, students, members of the organizing committee, and also of the Faculty of Mathematical Science, good afternoon. It's a truly exceptional honor to welcome you to the International Congress on Pure and Applied Mathematics in the Modern Era. Advanced challenges and applications in the contemporary world. Today, this virtual auditorium not only brings together brilliant minds, but also gathers decades of perseverance, boundless curiosity, and an unwavering commitment to the progress of humanity. The presence of our distinguished guests, whose names are already part of the history of science and thought, remind us that knowledge knows no boundaries. For the Faculty of Mathematical Science of the 400-year-old National University of San Marcos, hosting you is not merely a formal privilege. It's a reminder of our fundamental mission to foster dialogue, innovation, and the pursuit of truth. May this Congress be a space where ideas flow freely and where the inspiration of your achievements encourage new generations to look beyond the obvious. Thank you for joining us and for sharing your knowledge with us. You are all most welcome. I hereby officially declare open the International Congress on Pure and Applied Mathematics in the Modern Era, Advances, Challenges, and Application in the Contemporary World. Next, it's an honor to warmly welcome to PhD Carl Wieman, Nobel laureate in physics, who joined us today. We sincerely appreciate his presence and willingness to share his knowledge with us.